Welcome to Microlab Mastery. Today's topic, the streak plate method of isolation. The purpose is to obtain isolated or individual colonies from an inoculum. Colonies are isolated if they are not touching any other colony. During streaking, cell density decreases, leading to isolated colonies. One common method is the quadrant streak plate. This allows isolation of individual organisms from a mixed culture. In other words, a culture containing two or more species. To perform the quadrant streak plate, you will streak the culture back and forth on 1/4 of the Petri plate. You will then perform another streak with a loop that has been sterilized, beginning at one end of the first streak pattern. When doing so, be sure to overlap the first streak section only a couple of times. After sterilizing the loop, perform a third streak in a similar method to how you perform the second streak. Finally, after sterilizing, perform a fourth streak similar to how the third streak was performed. When the quadrant streak is correctly performed, you will find the concentration of the bacteria decreasing in each of the quadrants until by the fourth quadrant you're able to isolate individual colonies. To perform the quadrant streak plate lab, you're going to need the following materials: sterile nutrient agar plates, a permanent marker, bacteria cultures, and materials for aseptic technique, for example, an inoculating loop and a Bunsen burner. To perform the quadrant streak plate, please begin by labeling your plates. When you label plates, please note you label the base or bottom of the plate, not the lid. On your plate, please write your initials, your professor's last name, the class day and time, the table number you're sitting at, the name of the bacteria, and the temperature that you will be incubating these bacteria at. Next, please mix your bacteria culture. Do so by performing one of two methods, either flicking the tubes of the broth or by using a vortex. Next, we will be aseptically transferring bacteria from the culture to a microfuge tube. Ensure that you have a sterile transfer pipette and sterile microfuge tube to use. Ensure you are following proper aseptic technique by flaming the mouth of the culture tube after you remove the cap. Be sure you properly dispose of the transfer pipette by placing it into the biohazard bag. Then repeat these steps to aseptically transfer the second bacteria from culture into the same microfuge tube. Next, Please follow aseptic technique to sterilize a wire loop, as is being demonstrated here. After you sterilize the wire loop, please ensure that the loop is cool before you place into culture in order to avoid killing the bacteria. Aseptically transfer bacteria from the microfuge tube culture to the plate and streak back and forth in the first quadrant, as was previously demonstrated. Re-sterilize the wire loop and let it cool. After the loop is cooled, rotate the plate 90 degrees and perform the streak of the second quadrant, beginning at one end of the first streak pattern. When doing so, be sure to overlap the first streak section only a couple of times. After sterilizing the loop, perform a third streak in a similar method to how you performed the second streak. Finally, after sterilizing, perform a fourth streak similar to how the third streak was performed. Then ensure that you re-sterilize the loop. Please properly dispose in the microfuge tube in the biohazard bag. When properly performed, and when held up to the appropriate light, you may see faint streaks on the plate, as is demonstrated here. An alternative method you might be asked to perform is known as a zigzag inoculation. To do so, please divide up your plate into 4 sections using a Sharpie. Then, following the techniques described for the quadrant streak method, inoculate one of these 4 sections. If so desired, you could then use the other 3 sections for 3 different types of bacteria. If the concentration of the starting bacteria was dilute enough, it is possible you could get colony isolation using this method. However, it is much more reliable to use the quadrant streak method. This video has been a collaborative effort by Dr. Brian Sage and Dr. Dheeraj Verma. Until next time, this has been Microlab Mastery.